Dry age-related macular degeneration, (AMD), particularly its advanced stage, geographic atrophy, (GA), is a leading cause of irreversible vision loss in the elderly. From a medical affairs perspective, the introduction of Eyecyte-RPE represents a paradigm shift. It is a regenerative medicine candidate with the potential to alter disease progression, improve patient outcomes, and meet significant unmet needs in the ophthalmology landscape. I'm here today to provide an overview of this innovative therapy, its key innovations and clinical relevance, as well as the exciting opportunities that lie ahead. First, let's delve into what is Eyecyte-RPE. It is an allogeneic retinal pigment epithelium, RPE cell therapy derived from induced pluripotent stem cells, iPSCs, that has shown promise in preclinical studies for the treatment of geographic atrophy, a severe form of dry AMD. The cells are produced using a scalable and well-controlled process, cryopreserved at a single-use dose and formulated as a suspension product for ease of subretinal delivery. This approach holds the promise of providing a readily available, consistent, and effective therapeutic option for patients in need. The development of Eyecyte-RPE has been supported by comprehensive preclinical validation, including rigorous safety and efficacy studies. These studies have demonstrated no tumorigenicity or other adverse effects, as well as no immune-related adverse effects in non-human primates. Moreover, efficacy studies have shown significant improvements in visual function, further supported by histologic and electrophysiologic data. These robust preclinical findings have paved the way for approval to initiate human clinical trials, a pivotal step towards realizing its therapeutic potential. In terms of commercial viability, the market opportunity for Eyecyte-RPE is vast. With approximately 28 million people worldwide affected by geographic atrophy, the demand for effective therapies is substantial. Currently, there are no curative options available, making this unmet clinical need even more critical. Furthermore, aging populations in both developed and emerging markets are driving the incidence of AMD, thereby expanding the patient population and the need for innovative interventions. From a differentiated value proposition perspective, unlike autologous cell therapies, the allogeneic nature of Eyecyte-RPE enables mass production, a ready supply, a lower cost per dose, and broad access regardless of healthcare system. This makes it a compelling choice for patients, clinicians, and payers alike. Looking ahead, strategic considerations for medical affairs are multifaceted. Firstly, evidence generation and scientific communication will be crucial to articulate the clinical value of Eyecyte-RPE. This includes supporting data dissemination through publications and presentations and enabling peer-to-peer scientific exchange. Secondly, health economics and market access. Close collaboration with market access teams will be essential to develop pharmacoeconomic models and real-world evidence to support payer discussions and reimbursement strategies. Thirdly, KOL engagement and education. Building strong relationships with key opinion leaders and ophthalmology societies will be instrumental in facilitating early adoption and clinical trial enrollment. Finally, post-marketing surveillance and life cycle strategy. Planning for long-term safety monitoring and post-marketing studies is crucial. Additionally, exploring potential label expansion into earlier stages of AMD or combination therapies should be considered. In conclusion, Eyecyte-RPE represents a beacon of innovation in the field of ophthalmology. Its potential to redefine the treatment landscape for dry AMD is immense. With clinical trials underway, this strong translational foundation, it presents a compelling opportunity for strategic investment and cross-functional engagement. Embracing this innovation will ensure that patients have access to this transformative therapy, ultimately bringing hope where there was none before.